So our next speaker is Miriam Bornhorst from Lurie Children's Hospital of Chicago, who will be speaking on metabolic profiling in neurofibromatosis type 1.
Let's try and stand. All right, I have to apologize. My world is literally spinning. I've been having vertigo since I got here last night, so I am going to apologize in advance if I fall over. I'm just kidding. Perfect. Okay. All right. So I'm going to use this one. Okay, perfect. So I do have to disclose that I do some consulting with Alexia and as well as SpringWorks, but nothing related to this talk. So I want to talk a little bit about metabolic presentation in NF1. So neurofibromatosis type 1 is one of the RASopathies. So it is caused by an alteration in the NF1 gene, which is really important. It's a tumor suppressor gene. It's really important for kind of keeping signals or sort of regulating signals that are trying to get through that RAS-MAP kinase pathway. And so if you have an alteration in the gene, it makes it so that signal gets through when it's not necessarily supposed to, similar to many of the other RASopathies. And so with NF1, you have a risk for a lot of different things, tumors, for example, and then cognitive changes as well as some other problems. But one of the things that I'm really interested in is kind of this metabolic presentation of NF1, which is not something that's been talked about very much, but something that I think is very important for us to recognize. So there have been some studies done before looking at NF1 and sort of how patients with NF1 present in terms of their metabolic abnormalities. And some of the things that they found is that patients with NF1 have decreased bone mineral density, which puts you at risk for fractures as well as scoliosis. You also have decreased muscle bulk and strength. So when you have NF1, a lot of times we'll see gross motor developmental delays, decreased tone, difficulty with physical activities. And people have actually tried to look and see why are we seeing, or why is this phenotype there? And they'll find that within the muscle fibers, there's actually accumulation of lipids. And if you treat with very specific diets, so high medium chain fatty acid, low long chain fatty acid diets, you can actually decrease that amount of lipid accumulation in the muscle and improve on muscle strength in a mouse model. You can also see changes in BMI with individuals who have NF1, as well as decreased heights and lengths. So this is just a picture here just to show you sort of what I'm talking about in terms of the growth pattern that we'll see with NF1. So this is a patient, a female patient, and you can see here that the height and weight are both pretty consistently below the growth curve. Now, moving along that growth curve, and this patient had growth hormone checked and a lot of other things checked, and it wasn't related to a hormone problem. It's just this is sort of where they were in terms of their pattern. And then this is a male patient here. So again, on the left hand there you can see that the weight is really kind of on that lower side. Now the stature, if you look at that stature, it actually looks pretty good. So it doesn't look so bad. But pay attention to kind of this part here, right around 14 years of age, 16 years of age. The patient went through puberty early and then they peaked. So what's going to happen with this patient is that over time, their height percentile is actually going to decrease because they're no longer growing. So they do or will end up having a lower height as an adult. So what are some of the other studies that have been done looking at metabolism? So they've also looked at diet. So patients with NF1 actually are more interested in eating diets that are high in fatty acids and lipids. So they tend to have very fatty diets, yet they have very low weights despite that. Patients with NF1 also tend to have low glucose levels. So if you were just to take and look at glucose levels in a patient with NF1, you would find that those levels tend to be on the lower side. And then we also see increased insulin sensitivity in the patients. You can also see abnormal metabolism and changes in indirect calorimetry. So indirect calorimetry is a way for you to kind of measure energy expenditure. And so what we'll see with our patients with NF1 is that just sitting, they will use more energy. And I've actually looked at a cohort of patients with NF1 myself, and I can see anywhere from just a little bit over what would be normal to 360% more energy usage just by sitting in these patients. We also see a change in the respiratory quotient. And so that is something that tells us what type of energy source is that patient using. And usually for most of us, if you've just eaten, your energy source is carbohydrates. But for my patients with NF1, and they've shown this in other studies as well, you'll see that they actually oxidize lipids and fewer carbohydrates, even again in that quote unquote fed state. And then finally, one of the things that I think is very interesting is that when NF1, the higher plexiform neurofibroma tumor burden that you have, the more likely it is that you're going to have this metabolic phenotype. So I did a collaboration with Catherine Lenberg, who's at the Johns Hopkins University, and we looked at patients with plexiform neurofibromas with NF1, and we tried to look to see if there was any sort of a relationship between the plexiform neurofibroma burden and this quote-unquote metabolic phenotype. For this particular manuscript, we were looking mostly at heights and weights. And what you can see here is that basically the higher plexiform neurofibroma burden you have, so the further you go to the right on these charts, the lower your height tends to be, as well as the lower your weight tends to be. So there definitely is an association there. And I do just want to throw out too that we included some patients with NF1 microdeletions in this analysis. Patients with NF1 microdeletions do tend to have higher weights and actually higher heights just based on the way that that gene is deleted. And so at some point we should probably pull those patients out because I think that this association would be even stronger. So thinking about NF1 and again, RASopathies, and we've heard a lot now today already about MEK inhibitors. What happens if you inhibit that pathway, that RAS-MAP kinase pathway? So again, you have a change in NF1, the pathway is overactive. If we stop that overactivity, can it actually change this phenotype or does it change this phenotype? And what I'm showing here is some patients that were treated with MEK inhibitors. And you can see sort of, especially on the left-hand side, I don't have a lot of points there, but when this patient started on a MEK inhibitor, within months, their weight went up considerably. And then on this part here, you can see again, tracking along a curve very nicely. As soon as this patient started on a MEK inhibitor, we saw an increased trajectory in that weight. So then I took a cohort of patients with NF1 who were on MEK inhibitor and a cohort of patients of NF1 who were not on a MEK inhibitor. And what you can see here is that just kind of comparing those two cohorts together, the patients who were on the MEK inhibitor definitely had an increased weight percentile compared to those that were not. So then I wanted to look at this a little bit further. And, as you've heard here again today, sometimes there's things that you can't really study well in a patient. So I wanted to then look at mice to see how we can better understand this sort of metabolic phenotype. There are two mouse models that I used. One is a human GFAP-CRE mouse, which targets cells actually in the brain, but this human GFAP-CRE also targets cells in the gut. So we do definitely know that at least the intestines are influenced. And then there's a periostin-CRE mouse model that I used too. These mice form plexiform neurofibromas, but as far as we know, the CRE does not target anything outside of the heart and the nerves. So what do we see in these mice? So when we look at the mice in general, and so there's going to be four curves here. The curves are going to show basically a mouse that has NF1 deficiency, which is labeled as MUT or quote unquote mutant, a mouse that does not have NF1 deficiency, which would be our control. And then we have those that were treated with a MEK inhibitor, which in this case was the PD0325901, which now is called mirdametinib. And then the mice that were not treated with a MEK inhibitor, so your vehicles. And what you can see on this curve here basically is that the mice that were untreated that had NF1 deficiency had the lowest weights. And then those that were treated with the MEK inhibitor, so the mice that had NF1 deficiency that were treated with the MEK inhibitor had slightly higher weights, and then the controls were a little bit above that. And then if you put a control actually on a MEK inhibitor, their weight goes up pretty considerably, particularly in the males. So there definitely is an impact on this MEK inhibitor, again, on the weights in the mice, similar to what we were seeing in our patient population. So then I looked at the metabolites that are involved, and I tried to figure out, okay, so how is it then that the MEK inhibitors are influencing weight? And what I was sort of expecting to see is that there probably was sort of an impact on that fatty acid sort of metabolism pathway. And sure enough, when you look at all of the different metabolites that are involved after MEK inhibitor treatment or with MEK inhibitor treatment, the ones that were the most significant on the top there in terms of increase were those that were related to fatty acid metabolism. So there definitely is a role there. I do want to kind of point out, you can see serotonin at the very top there. I still don't really understand why, when you're on a MEK inhibitor, your serotonin levels go from like 0 to really crazy high. But I did have a student look at this with me, and she actually found that when you have NF1 deficiency, your tryptophan levels are low, which is an amino acid that's needed to develop or to create serotonin. And then when you are on a MEK inhibitor, those levels actually go to normal. So we think that it might be related to that. So when we look at this just a little bit further, breaking down this, again, fatty acid metabolism pathway, you can see here that, I mean, there's a lot of stuff on this slide, but basically what this is showing on the left side there is that I had within the mouse model, mice that were not treated and mice after treatment, and we saw very significant, anything that's green on there is a decrease. So we saw a decrease in your fatty acid metabolism, in the fatty acids or free fatty acids in those mice. And then what it is, I looked at the humans or the patients that I had, and I saw a very, very similar pattern. So again, patients that were not on MEK inhibitor compared to patients that were on MEK inhibitor, we saw a decrease again in fatty acids. And so then we took it one step further. Unfortunately, I didn't have enough sample for the mice to be able to do this, but for the patients, I was able to send it off for lipidomics, which is looking more so at your triglycerides and some of the lipids in the body. And what we saw is that after MEK inhibitor treatment, there was an increase in the triacylglycerol esters, as well as a decrease in your androgenic steroids. So what does this mean? All of this data that I just showed you. So basically, in summary, when you have mice or patient samples, they both showed a decrease in your long-chain fatty acids or your fatty acids after MEK inhibitor therapy. And you can see a similar trend in patients and mice in terms of the median chain fatty acids. The decrease in long-chain fatty acids accompanied by an increase in triacylglycerol basically tells us that there's a decrease in lipid oxidation, which could potentially account for increased weight gain in treatment. So you're not oxidizing those fatty acids as much, you're actually accumulating them and or storing them when you're treated. And so that's why these patients, as well as the mice, probably gain weight. And then I think it's also important, those androgenic steroids, it's also important to mention that that decrease in androgenic steroids, so steroids are important for increasing muscle mass and decreasing fat mass. And so when you're on a MEK inhibitor, when you have an increase in that, it actually will decrease your muscle mass and increase your fat mass. And so that might also help to kind of explain why these patients are gaining weight. But that decrease in muscle mass, I think maybe could be related to why we're seeing this elevated creatine kinase in patients who are on MEK inhibitors. So I just wanted to show one more sort of study that we did on our mouse models, and this is something that we're extending now. As I told you, with patients with NF1, they have an increase in their energy expenditure. And so what we saw actually in our mice, we looked at males more so than females because they had a stronger phenotype, is that the male mice that have NF1 deficiency do tend to definitely have an increase in their energy expenditure. And then when we look at insulin sensitivity, as has already been described in our patients, we're also seeing this prolonged insulin sensitivity in the NF1 deficient mice. But interestingly enough, we also see that in the MEK inhibitor treated control. And so somehow the MEK inhibitor seems to be influencing your insulin sensitivity. And that's something I'm trying to look into a little bit further. So finally, in conclusion, again, we know that patients with NF1 in general tend to have decreased weight, lower heights, and decreased muscle strength or bone tone, and then reports of fatigue. And then previous studies have shown abnormal fat accumulation and muscle biopsy. And so the studies that I have done have shown with between mice and patients that MEK inhibitors can potentially increase your lipid oxidation, decrease lipid oxidation, which then increases the accumulation of fat in those patients. And so if you flip it backwards, what that tells you basically is that when patients have NF1, they're using fat more so for their energy source than they should be. And so where am I going to go next with this? There's a lot of ways that I can take this. I think that it's really important for us to better understand why it is that patients with NF1 or other RASopathies have sort of these alterations in their energy usage. I think it's really important for us to also do additional studies, particularly in patients, to better understand this. But then I think also, again, using the mouse model, most of what I've done so far is using blood. What's happening in the muscle, what's happening in the heart, what's happening in the liver. So not only looking at those blood, but also looking directly at the organs to see what's going on. I'm like looking at biomarkers, so things that could potentially tell me, is there something that we can use this for in terms of, your metabolic profile, telling you who's at risk for developing tumors. And then finally, I think what's going to be most interesting, especially for NF1, is I'm doing a number of studies now in the lab where I'm trying to see if you make the environment so that it's quote unquote toxic to tumors, can we potentially decrease that risk for developing tumors? This is just a slide acknowledging collaborators as well as people in my lab who've helped with this project, and then my funding sources. Thank you.
Great. Thank you so much. Really interesting talk. We have time for some questions. No?
Elliot Robinson, Cincinnati Children's. Wonderful talk. So, I know that many people are looking at mitochondrial function and the concept of RASopathies. And I know Bob Kesterson showed at the NF meeting in the CAG-CRE NF1 knockout model that there was mitochondrial dysfunction. How does these metabolic alterations kind of fit with this, with what is emerging in terms of, abnormalities in mitochondrial, energy production and whatnot?
Yeah, no, I think that's really a good question. And I know that he has been definitely looking into that a lot. I haven't actually looked into it myself, so it's hard for me to say how that relates to what I'm seeing. Really, what I'm looking at right now is more of a global approach to sort of your metabolic profiling, or a global approach to what's happening with metabolism, and that's more cellular. And so I think once I start looking more so at the tissues, then I can hopefully be able to correlate the two. But right now, I'm not able to comment on that.
Other question?
Yeah, I'm Gorieli Oxford. Do you want to try to tell us what's different about the microdeletion? Because biologically, that doesn't necessarily make a lot of sense.
Yeah, so patients that have a microdeletion, so not only is the NF1 gene deleted out, but there are genes that sort of flank that NF1 gene, and oftentimes those are also deleted out. So depending on the size of the microdeletion, you can have, sort of upstream or downstream genes that are deleted out along with that NF1 gene. And we think that it's not necessarily the NF1 gene being deleted, but it's probably related to the other genes that are deleted out instead. Yeah.
Over here, Antoine Bennett, Yale Medical School. Really interesting talk. I was wondering whether you had thought about, or whether you'd looked about looking at muscle composition in these, at least in the mouse model, in terms of whether there's a shift in oxidative glycolytic composition, which would go a long way in terms of explaining the energy shift that you're seeing.
Yeah, definitely. Yep. So we have been collecting muscle samples, and that is definitely something that I'm hoping to look at, not only again in terms of the metabolic profile, so sending it off and trying to figure out, what pathways are being used in the muscle, what does that metabolism look like within the muscle. But I think, like I told you, that somebody had already looked at muscle and they showed, sort of accumulation of some fats within the muscle. I'd also like to look at it a little bit closer to see what sort of fat accumulation is occurring, particularly in our mouse models that because the mouse model that they were using actually had a Cree that affected the muscle, right? So, but my mice, or the mice that I'm using, we are also seeing decreases in the fat, decreasing the fat storage, but also changes in the muscle too. And so I'd like to see, for those that are not necessarily Cree specific, what we're seeing in the muscle. So yes, that is something definitely interested in doing.
So I'd like to actually dovetail off of that excellent question with regards to the skeletal muscle, because it's the exact opposite of what we saw in our Costello and CFC
Oh,
model
interesting.
with the MEK inhibitor.
Huh.
So how did you assess the skeletal muscle showing that it actually was a decrease in mass? Did you look at fiber type? Did you look at, with regards to the myofiber size? Did you look at the belly of the muscle, the whole muscle itself, like weigh the triceps or
Yeah.
weigh something like that? So how did you assess that? Because it's the opposite of what we saw.
Yeah, so I have to just go back for a second and apologize. I didn't actually look specifically at that yet. It's more of a theory based on the androgenic steroid, or so the level of the androgenic steroids within the plasma, and what you'd sort of expect to see with that. But yes, definitely do need to look at that closer. Yeah.
Because we actually saw with the MEK inhibitor that we normalized muscle at both the histological level as well as the functional level, and as well as the actual weight of the muscle normalized to control.
Yeah, that's great. That's great. Yeah, something definitely interesting to look at, because I know with NF1, when patients are on MEK inhibitors, they do tend to complain about feeling a little bit weaker. And I've personally had to take off, patients from MEK inhibitors because they had such significant decrease in their muscle strength that they would lose the ability to hold up their head, for example, or lose the ability to even walk. And so I
That's really
wonder if there's a
interesting.
difference in how the NF1 patients respond to the MEK inhibitor.
So where do you think the weight came from the patients being treated with a MEK inhibitor with the plexiforms? Where the subcutaneous tissue, fat?
Yep, a lot of it sits right in the neck area. And it's interesting because in the mice, when I treat them too, they get this massive fat pad, like right along the shoulders.
Kinda like
Some
a-
in the stomach too, but
Cushianoid.
yeah, very similar.
Okay,
Yeah, very
very
similar.
interesting.
Yeah.
Okay,
So,
thank you
thank you.
so much. Fascinating. Oh, sorry, there we go.
Oh, thank you.
Yeah.
Alessandro De Luca from Rome. I was wondering whether you considered to include in your study also, there are a few variants in the NF1 gene, which are missense mutations that predispose more to like a RASopathy-like form of NF1, like in neurofibromas, but having more RASopathy-like phenotypes, like short stages, macrocephaly, and heart disease and dysmorphism. And that could be interesting.
Yeah, that would be very interesting. And it's a really good suggestion. I mean, right now I'm just using sort of the standard model where you have the deletion of the exons in NF1. But if you could actually do point or, variant specific models, I think that would also help us really understand this better. That's a really great point. Thank you.
Oh, one quick last question. The discussion's really good.
From a parent perspective, actually, full disclosure, my daughter is a patient. I didn't even know you were going to be here, but I'm just curious about the implications for the broader NF community with these. We're looking into, even just the types of fatty acids that children should, people should be eating.
Yeah.
Like, does this have broader implications for lifestyle choices that could possibly influence and help people dealing with NF1?
Yeah, so I'm hoping it will. So right now, when parents ask me, what can I feed my child to, prevent tumors or to help them feel healthier? I don't actually have an answer to give yet, which is why I'm starting to really look now at different diet interventions, particularly with the mouse model. And I'm trying to see what we could potentially do to help with some of the phenotype that we're seeing in NF1. And again, I think if we can find a diet or some sort of a change that you can implement, as a parent, I think that this would be something really strong, even early on in the child's life. And it's nothing's ever going to be perfect. Like people always say, well, you can't treat something with a diet. And I get that, but I think that it definitely would potentially help the sort of outcomes or help that quality of life for patients with NF1 and potentially even other RASopathies, depending on how this kind of plays out. So yes, I am hoping someday to have an answer to that, just not quite there yet.
So adding medium chains, medium chain fats?
Medium chain fatty acids have already been shown to improve muscle strength and muscle tone in a mouse model. And then they're kind of looking at it right now in patients too. So it's Mediterranean diet type of a diet. It's something that you could potentially try. But yeah, I do think that there's even ways that we could expand on that and hopefully even improve on that.
So from a biochemical perspective, patients that we see in the clinic that have respiratory chain, we were talking about the mitochondria, adding medium chains to the diet actually really helped.
Yeah, I believe it. Yep. Great. Thank you.
Thank you so much.